So this is the second embryology teaching that I have created and teaching myself. Very proud. These are some nice sperm cells that we've got on the side. I apologize, I've got a bit carried away with these and they're on most slides because I thought they were really cute. So just to cover the learning objective, first of all, we're going to be discussing the process of neural tube formation, including the main 4 steps involved. We are going to define and describe the process of somite formation and their differentiation. We're going to cover the basics of the embryonic development of the kidney and the gonads, the general derivatives of the mesoderm, and the process of embryo folding, and the major consequences of this event. So hopefully this goes ahead well. We're going to just do a quick recap to start with. I know some of you guys will have been in the first embryology one and some of you won't, but if you just give it your best go. So if you go, you can either text this or join the link to the poll everywhere. Hopefully this works. Can you guys now see this poll everywhere screen? Can one of you just say yes or nod? If you can see a poll everywhere screen. No. This is annoying. I don't know how to. Ah, here we go. Okay, great. Can you guys see that now? Great. So if I activate that, you guys should be able to do that. So at what day does implantation of the embryo occur? Oh, some people are changing their minds. Okay, so it is day 8 to 9. So 20% of you guys got that. The next one. What is the decidual reaction? Oh, it's quite mixed, to be fair. Okay, so the correct answer is a thickening of the endometrium around the gestational sac. So well done to the 10% of you that got that. The next one, what structure is considered the primordium of the central nervous system, or like the precursor? Yeah, I think most of you got that. So the neural tube, the neural crest cells are the precursor for the peripheral nervous system. I think this should be the last one. So what is the completion of implantation marked by? Yeah, I think most of you got that, the fibrin plug. And, oh no, there's one more. How many cells comprise a morula and at what day does it form? So 16 cells and it forms at day 3. Cool, I think that's everything. So if I get cracking, can you guys see my PowerPoint again? No. Resume share. There we go, share. Can you see it now? Cool, perfect. So we'll get started. So this is a really key figure for you guys to know. So if you take away anything from this lecture, try to remember as much as this as possible. It's really useful. So we'll cover it in more detail later on in the talk. But basically you have the endoderm, the mesoderm, and the ectoderm, which we're going to focus on for now. So the ectoderm splits into the surface ectoderm and the neuroectoderm. So the surface ectoderm is everything on your surface, basically. So your skin, your hair, your nails, the enamel on your teeth. And your neuroectoderm can be subdivided into your neural tube, which, as we've just said, is a kind of precursor of the central nervous system, and some additional stuff like the retina, posterior part of the pituitary gland, and the neural crest cells are the precursor of the peripheral nervous system. So that's really important to remember. The endodermal derivatives include the epithelial lining, so of the trachea, the bronchi, the lungs, the lining of the internal organs, including those in the GI tract, and the renal and the UG system. And then the mesoderm derivatives can be subdivided into 3 components. So you have the lateral mesoderm, the intermediate mesoderm, and the paraxial mesoderm. So these give rise to things like connective tissue, so ligaments, tendons, aponeurosis, muscles, so smooth muscle, cardiac and skeletal, blood, lymph, cardiovascular organs, spleen, and so on. So I will put these slides on the pediatrics Facebook page at a later point on a Google Drive as well, so you guys can access these. So we're going to first talk about the process of neurulation, which is just the formation of the neural tube. And as we've discussed, that's a precursor of the whole of the central nervous system. So there's 4 main stages. So we have the transformation of the general ectoderm into a thickened neural plate, and you can see this on the figure on the right. The neural plate becomes shaped, so it becomes narrower and longer. And then we have a lateral folding of the edges of the neural plate, and these are then obviously called the neural folds. And finally, to become a tube, we need to have some kind of fusion event. So the 2 lateral apical surfaces of the neural folds fuse, and then they become a neural tube. So we'll go into that in a bit more detail now, but that's just a basic overview. So this process occurs at days 18 to 21. And around day 18, so at the start of it, the neural plate invaginates along its central axis to form the neural groove. So you can see on these figures above, you have the notochord in red running along the bottom, and you have the neural plate that forms along the top of it. And you can see in the middle image, this then starts to invaginate in the middle, which is the groove, and you've got the neural plates on the edge. And this also starts to form the mesoderm. So you can see the mesoderm that forms within this middle layer here. And you can see this then splits into the lateral mesoderm on the outer edges laterally. You've got the notochord that's formed now in the center, and you've got the paraxial mesoderm there as well. So this is a nice image which just shows it in real time, basically. So you can see the neural plate there, and you can see the thickening of the neural groove down the central line there. And we'll come to this at a later point in the lecture, but there's somites you can see on this image here that form alongside this neural groove as well. So yeah, like I said, it begins at the start of the 3rd week and it's completed during the 4th week. The masses of cells attach themselves from the lateral side of the neural plate and form the neural crest cells, which, like I've said, give rise to the peripheral nervous system. The end of the 3rd week, the neural folds have begun to move together and fuse, which turns this into a tube. And the neural tube then separates from the surface ectoderm. The surface ectoderm fuses, and then that differentiates into the epidermis of the skin. So you can see that's what's going on in this image here. You've got the neural crest cells in green by the side, and then they subsequently form over the top of the tube. So Neuropore closure. So we have a anterior and a posterior end of the tube called the anterior and posterior neuropores. And it's quite self-explanatory. The neural tube is formed when these neuropores close. Before they fuse, the cephalic, so the head end, and the caudal, the tail end, communicate with each other via the way of the amniotic cavity. So the anterior neuropore closes around day 25 and the posterior one slightly later at day 28. And once the neuropores have closed, particularly the caudal neuropore, the embryo has a very basic spinal cord and brain vesicles. So it's the start of the formation of the central nervous system. This is quite a nice image I found, and it just basically shows the development of the central nervous system. So we've got the neural tube, and we've got the start of the development of the brain now. So we've got the proencephalon, mesencephalon, and rhombencephalon, and then we've also got the spinal cords. And you can see on this image that these then subdivide into further structures. And then you can see by week 6, we've got pretty good development at this point of particular regions of the brain. So we've got the thalamus, infundibulum, tegmentum, pons, cerebellum, and so on. Like I say, these will be online for you guys to get as well. So somite development. So at the start of week 3, the paraxial mesoderm, so if you remember, we have 3 types of mesoderm. The paraxial mesoderm is organized into segments. They're quite rudimentary, so early on called somatomeres, and then by the end of week 3, so quite quickly, these then further organize into somites. So you can see on the image of the right, the little ball-looking structures of the somites that form along the central axis there. Also, they develop at a rate of around 3 pairs per day. And by the end of the 5th week, there's about 44 of them present. So they then differentiate. So various regions of somites along the edges of the neural tube become committed to forming particular cell types. So they all look identical if you were to look at them under a microscope, but depending where they are on that central axis, kind of confines them to a particular cell type. So they're transient structures, but they're absolutely essential in the development of the body plan in vertebrates. So it's essential for correct segmentation, bone and muscle development, as well as creating the kind of correct template for the nervous system. So the different cell types include the dermomyotome, and this is a double layered structure, and that's comprised of both the dermatome and the myotome. So again, self-explanatory, if you just remember dermomyotome, you remember the 2. The derma, like is in skin, it generates the dermis of the back, and the myotome is a lower level of the 2 layers of the dermomyotome, and it produces the myoblasts, which give rise to the axial and appendicular or limb skeletal muscle. And then we have a 3rd one, which is a sclerotome, and that's located of the ventral medial region of the somites along the central axis. And that gives rise to cells such as cartilage cells or chondrocytes, vertebrae and ribs, bone components of the axial skeleton, so rib cage, sternum, and so on. And you can see in this image down here below, it's quite hard to determine what's going on, but you can see we've got the neural tube in blue there, and then we've got some of the somites going on the sclerotome. You can see, particularly in the 3rd image along, you can kind of see the differentiation of the different layers. Again, this is just a nice summary image. So as I say, the somites come from the paraxial mesoderm, and that's called somatogenesis. And then you can see we've got the dermomyotome with the dermatome and the myotome, and the sclerotome in the ventral medial region, giving rise to tendons, cartilage, and so on. Basically all what I've just said. So dermatomes, you guys will have heard of. So it's defined as an area of the skin in which a sensory nerve derived from a single nerve root. So unfortunately, this is just a case of learning where's where. I've attached a really nice, quite amusing YouTube clip for you guys to watch in your free time called the Dermatome Dance, which is basically just, there's quite a few versions. It's a crazy woman that's going around, and she's, as Jed's doing it for us, it's a crazy woman jumping around, and she's basically painted herself with the dermatomes, and she does a little dance, and it's quite a good way to remember it by. Obviously, if you have pathology in one of these regions, you can then link it back to its dermatomal region and what its innervation. So really important. So moving on to embryonic development of the urinary system. So the urogenital system arises from the intermediate mesoderm this time. And the intermediate mesoderm forms something called a urogenital ridge, which, as you can see in this image on the right, you've got the, let me just move this out of the way, the neural tube in the center there in the blue, and then you've got the somites or dermatomes in the pink. You've got the big yolk sac in the yellow, and then you can see where it's labeled number 4, you've got a nephrogenic cord. So that separates the paraxial and lateral mesoderm, which is really important. And the nephrogenic cord is just a part of the urogenital ridge, and it's basically responsible for giving rise to most of the urinary system. So the nephrogenic cord then goes on to develop the pronephros, the mesonephros, and the metanephros, which we'll go on to discuss now. So the pronephros starts forming in the 4th week, and it's the earliest of the 3 structures, and it's transitory and non-functional. So it develops and it starts in the cranial region. So again, if you look at this image on the right, it's pretty much developing right up towards the head of the embryo. And it divides into nephratomes. The pronephros forms the kidney in the larval stages of fish and amphibians, but it is, like I say, not functional in humans, and it basically atrophies and just goes away. It is succeeded by the mesonephros, which we'll discuss in a second. And a good way of thinking about it is like a head kidney. So it develops, like I say, in the head region of the embryo. So then we move on to the metanephros. So this differentiates during week 4, and it basically just takes over from the pronephros. So again, we can see on this image, it's developing further down the central axis of the embryo now. And it's classed as a first excretory organ. So it's like a very, very rudimentary kidney, basically. And it develops from 3 structures. So we've got the nephrogenic cord, as I've discussed. We've got the mesonephric ducts and the glomerular capillary network. And because it's excretory, it starts a production of urine at week 6. And after the 10th week, it basically becomes inactive and atrophies again. This is because it's taken over by the metanephros, which is the functioning excretory kidney, and this basically persists in adults. So this is what we all end up with, and it's not taken over by anything else. So this starts development during week 5, and it forms from the uteric bud and the metanephric blastema. So like I say, this persists as an adult, and the metanephric blastema induces the branching of the ureteric bud, which gives rise to the kidney calyces and the collecting ducts, and basically forms a more developed kidney. So as you can see on the right here, at the start of week 5, we've got this metanephric duct in the purplish colour, and we've got the ureteric ducts or the bud in yellow. And the metanephros, as you can see, is composed of the ureteric bud and the metanephric blastema in the pink. Further development up till week 8, you can see it's growing, expanding, differentiating. The metanephric blastema becomes the kind of component or the body of the kidney, and we've got development of its components, so the renal pelvis, the major and minor calyces, and the collecting duct, and this persists as an adult. This is a nice summary slide again, so just showing kind of what I've already said. So we've got the 3 mesoderms, paraxial, which we've already discussed, the lateral and the intermediate, which is the basis of the urogenital system. We've got the urogenital ridge, which gives rise to the nephrogenic cord, and that in turn produces the pronephros, the mesonephros, and the metanephros. And it tells you kind of what's going on here. So the metanephros is composed, as we said, of the mesonephric ducts and the metanephric blastema, which is a uriforming system, and we've got the urine excreting system as well. So discussing the embryonic development of the reproductive system, we have the gonads, which are derived from 3 sources. We've got the intermediate mesoderm, as we've discussed, the underlying mesenchyme and primordial germ cells. The primordial germ cells appear at the time of gastrulation, and they migrate out of the embryo into the wall of the yolk sac. The primordial germ cells are basically just highly specialized cells that are precursors of gametes. So during week 6, we've got these primordial germ cells, which are incorporated into gonadal cords, and they will, like I've just said, eventually differentiate into the oocytes and the sperm cells. Bit of a complicated image going on the right here, but if you just look at the 2 furthest images on the right, you can see the very basic structure of both males and female genitalia systems in a newborn. We have genital ducts arising from the paired mesonephric and paramesonephric ducts. So if we remember, if you guys have covered it, we've got the Wolffian duct, which is in males. So I tend to think of it's a bit of a weird one, but males as wolves. It just helps me remember it. And we have the Mullerian ducts in females. So I think of again, really odd, it might just help you. I'm a female and I enjoy Mullerices. So I just remember Mullerian, Mullerice, female, bit of a strange one. So they give rise to the male and the female genital ducts respectively. Moving on to sexual differentiation. So week 7 now, we've got the start of the differentiation of the gonads and the reproductive tracts in males and females. Really important to remember, this is influenced by the SRY protein, or the sex determining region Y on the Y chromosome. So you can see that on the chromosome image over here. We've got the Y chromosome and we've got the sex determining region in the blue strip there. It needs to be active on the Y chromosome to induce sexual differentiation. And like I've just said, this determines gonadal differentiation, development of the internal ducts, external genitalia, and also gonadal descent, as well as brain sex. So you can see on this flow diagram, we've got the SRY protein, and we've got, this is in a male, so we've got the production of Leydig cells, which produce testosterone, and we've got the Sertoli cells, which are like a supportive cell in the testes, and they produce the anti-Mullerian hormones. So if you remember females, Mullerice, all that. We, as males, we want to produce anti-Mullerian, so we're not developing any female genitalia. And then we also have the indifferent gonad, which produces primitive sex cords. And then this leads to, say, the development of the testes in males. So we've got the seminiferous tubes and the rete testes as well. Obviously, if these go wrong, you can have various sexual differentiation disorders. So, for example, you can have agenesis or dysgenesis of the gonads. You can become under or over virilized. And an example of that is congenital adrenal hyperplasia, which is common. I think it's about 70% of sexual differentiation disorders. So moving on to general derivatives of the mesoderm. Again, this takes back to the first image, just in case we're trying to remember what goes where. So I found this, completely forgot the word for what these things are called now. They're a thing. If you just remember mesoderm, I found this on YouTube, it gives you everything that you need to remember what derives from mesoderm. So you've got the mesothelium, you've got all the muscles. E stands for nothing. There's nothing that goes in E, so it's just X. S for spleen, serous lining, somites. O for osseous tissue, ovaries and outer layer of the adrenal gland. D for the ducts, and the ducts of the genitalia, as we've just talked about. E for the endothelium, R for renal tissue, and another M for microglia, mesenchyme, and male gonads. So it's quite a good way of remembering that. Lastly, folding of the embryo. So up to this point, we have a flat trilaminar embryo, and we want to fold it into a cylindrical shape. And this starts at the beginning of week 4. So this folding process encloses the mesoderm within it and the endoderm, and we have the ectoderm on the outside, which, if we think ecto outside, becomes the skin. So we have 2 folding processes that occur at the same time. So we have longitudinal folding, or cephalodecaudal flexion, so head to tail, and transverse or lateral folding. And this leads to the formation of the abdominal wall in the embryo. So the abdominal wall comprises of the foregut, the hindgut, and the midgut, and they come from the folding of the endoderm cell germ layer even. Like I say, these occur at the same time, they're not separate events. And then as a result of this, the embryo increases rapidly in size along its vertical axis, because central growth at this point is greater than peripheral growth. So the dorsal region of the embryo grows more rapidly than the ventral region, and it causes the embryo to become like a curved shape. Yeah, so that's pretty much it. We've gone through that quite quick. Again, this is just a nice image. You can kind of see what I've discussed here. So you've got the cloacal membrane, which will go on to become the anal region, and the oropharyngeal membrane, which will become some of the structures within the throat and the mouth. You can see the folding planes in this image here. So we've got the longitudinal and the transverse plane. Yeah, these are some additional resources that I find useful that you guys might. So there's a really good embryology website, which it can be a bit heavy, but it's got so much content and information on. The second is a video, a guy, Amando, on YouTube, which has some really good embryology stuff. And Ninja Nerd, you guys have heard of it. He's got some really good YouTube lectures on embryology as well. So that's it. Hopefully you guys have taken something away from that. Don't forget to check out our Facebook for future events and Instagram and whatever. And I'll also put on these slides as well at some point, either later or tomorrow, so you guys have access to them.